Good morning, everyone. I'm Sandy Williams, president of the Gladstone Institutes, and it's my great pleasure today to open up this special day for Gladstone and for the entire scientific community. We're pleased again this year to host the Ogawa-Yamanaka Stem Cell Prize, and you'll be hearing more about the history of that and the process by which today's awardee was selected. So I want to welcome Gladstone scientists and staff and honored guests who are here with us in the auditorium, and I also want to welcome all of you who are watching the live video cam streaming of this. So it's my pleasure now to introduce Deepak Srivastava. Deepak directs cardiovascular research at Gladstone, as well as our Roddenberry Stem Cell Center. And he has chaired the prize committee and led the process that we're here to honor today. Deepak.
Thank you, Sandy, and I'd like to welcome all of you to the 2016 Ogawa-Yamanaka Prize lecture by my good friend Doug Melton, who I'll introduce in a moment. First, to say a few words about this prize. You'll have a chance to meet Mr. Ogawa in a moment, but this prize is really meant to recognize and promote scientists who are pushing translational efforts with cellular reprogramming technology for regenerative medicine. And it's particularly nice for Gladstone to be bestowing this award this year because this is, in fact, the 10-year anniversary of the cellular reprogramming discovery of iPS cells made by Shinya Yamanaka. And there's been a year-long celebration of this milestone event. There are a number of individuals that we have that have been involved in selection of this prize. Last year, the jury selected Masayo Takahashi from Japan for being the first person to introduce iPS-derived cells into humans, which was a milestone landmark event. The jury is made up of myself and Shinya, as well as George Daly from Harvard Medical School, who's the soon-to-be dean of the Harvard Medical School, Hideyuki Okano, who is a renowned stem cell biologist at Keio University and is also the dean of the Keio Medical School, and Marius Wernig, who has pioneered neural reprogramming locally here at Stanford. The prize for the 2017 prize, the nominations will open January 1st and extend through March 31st. So we encourage those of you in the audience and watching online to think about those dates and nominate candidates for the future. So I will say more about our honoree in a moment, but at this time I'd like to introduce Shinya Yamanaka to say a few words about this prize. And Shinya needs no introduction, but is an investigator here at Gladstone Institutes and also directs the CIRA Institute at the Kyoto University, where they have really pushed the translation of iPS technology. Shinya?
Thank you very much, Deepak. Good morning, everybody. I'm Shinya Yamanaka. So it was 2008 when I first met Hiro Ogawa and his wife, Betty. They gave us, Gladstone, a huge donation to support our AIDS research, which resulted in the generation of a new drug. Since then, Hiro and Betty have been a great mentor to me and to my family. And also, Hiro has been a great coach of my golf. I'm very grateful to him. 2 years ago, Betty and Hiro came to me in Kyoto in April. And he started talking about a prize. They insisted to name the new prize Ogawa-Yamanaka Prize. I insisted it should be Betty and Hiro Ogawa Prize. We argued. And I just say, okay, we should continue this argument next month, because I come to Gladstone every month, so I have a chance to see them every month. But very sadly, Betty passed away before I met her again. So I decided to accept the offer to have Ogawa-Yamanaka Award, because I heard it was a strong wish of Betty. I still remember Betty very well. They were kind enough to come to Stockholm to celebrate the Nobel Prize. So I really cannot thank you and Betty enough for supporting Gladstone. So please join me welcoming Hiro Ogawa. Hiro, please.
Thank you, Shinya. The way I felt is behind the scene of any successful scientist or managers, there are always somebody behind that is a spouse. And I know that some of our work without having a family's complete support, sometimes it's very difficult to achieve. And my wife and I was thinking, look at Chika and Shinya. And I can see that Chika was really supporting Shinya until he became famous. And truly, she was the main person who were keeping the Yamanaka family. 2 daughters, going to school, and she has nothing to do with those. She was just studying, you know? So anyway, we decided, okay, maybe we should do something that we can really bring the opportunity where the scientist or science can be clearly translated into the patient. Everyday patients, unfortunately, with no remedies available in current technology, but with the stem cell development, they are quite possible those people can be saved. And therefore, we are talking about, let's try to promote translations and applications of the science into the clinical. And that's what we wanted to promote. So Betty, my wife, and I proposed to Shinya, can we have such a prize which may encourage the world of a scientist to really come up with the quicker, earlier application to the patient so that the patient can be saved. And that's what we wanted. So on not only the scientific reason to promote, but also we wanted to make sure that this prize always appreciate a person behind the winner. Last year, Dr. S. Takahashi, I really appreciated to her husband for all the support. And I just talked to Doug this morning. Thank you very much for your acceptance of this prize. Next year, when the Stem Cell Institution of a Stem Cell Research Society will have an annual convention in Boston. That's the time when Chika and Shinya are going to be there, and we all want to invite you and your wife to a good Japanese restaurant. Again, thank you very much for this opportunity, And thanks again for the wonderful selection committee's decision. Thank you.
Thank you very much, Hiro. And for those of you who have a program, if you look on the back of the program, there's a photograph of Hiro and his wife, Betty, taken at the 2012 Nobel Prize ceremony in full-on in their white tie and formal attire. So we miss Betty a lot, but know that she would be very proud of what has transpired and what we're doing here today in her honor. So at this time, it's my pleasure to introduce our awardee today, Dr. Doug Melton. So Doug grew up in Illinois and went to University of Illinois in Champaign, and then decided to move to England for a while, where he was a Marshall Fellow at Cambridge, and there met Dr. John Gurdon, and proceeded to obtain his PhD degree there with John, which is quite fitting for this award. As all of you know, Dr. Gurdon shared the 2012 Nobel Prize in Physiology and Medicine with Shinya. And he was so successful there that after just completing his PhD, he was recruited to Harvard to start on as a faculty member without even doing a postdoc. And all the postdocs in the audience, I know, would be jealous of that. But he proceeded there then to really make some major findings related to the developmental biology of how cells are acquiring their fate. So Doug is really a developmental biologist, and I know all of us, those in the field, certainly in the 90s, when I entered the field, saw the work that he was doing as the most rigorous and really using, at that time, I think, frog embryos, largely really defining how cells are acquiring their fate at the earliest stages after fertilization. He subsequently, in the mid-90s, and he said it's okay for me to share this, had a son who was then diagnosed with diabetes. He quickly made the decision to focus his laboratory on not just understanding how those cells during embryogenesis acquired their fate, but more specifically how insulin-producing cells in the pancreas acquired their fate, and how he might be able to utilize that information from developmental biology to generate pancreatic insulin-producing cells for future therapies for diabetes. And he'll share with us his progress over the last 20 years in doing exactly that. He subsequently found a few years later that his daughter, who was older, also developed diabetes as an adolescent. And so this is a very personal mission for Dr. Melton, but he's brought to it the full heft of the most rigorous science to this. He's made many advances in this area, some of which he'll share with you, but he may not share that during the period that generation of human embryonic stem cells became quite difficult for various political reasons, Doug was the one who led an effort at Harvard to use philanthropic dollars and private dollars when federal dollars were not available to generate nearly 30 human embryonic stem cell lines that have really propelled the field forward at that time. He has since been able to efficiently make insulin-producing cells, and in keeping with the theme of this award, has really not just left the discoveries there, but pushed them towards translation, and very recently was able to get commercial investment for a startup company to really take this into clinical trials. Along the way, he's been served as co-director of the Harvard Stem Cell Institute and has been an investigator of the Howard Hughes Medical Institute. His accomplishments have been recognized in many different ways, including election to the National Academy of Sciences and the American Academy of Arts and Sciences. And we're very pleased internationally with the fact that Doug will be the incoming president of the International Society of Stem Cell Research, where he'll have an even broader platform to lead us in the stem cell field. What you don't know, probably, is that in addition to being a wonderful scientist and a wonderful father, as I mentioned, to his children and devoting his life towards that effort, he is a parent to many, many others. He is serves as a parent figure to 440 Harvard undergraduates by serving as the resident associate at Eliot House at Harvard, which is the dormitory, one of the dormitories there where he and his wife live in that building, dine with the students every day, and he's completely dedicated to undergraduate education, teaching classes, and we're lucky that he's agreed to come here today because Doug travels very little, in large part because he's always teaching, and that is the most important thing for him. So I think I wanted to give you a flavor for Doug as a person. He's also an avid tennis player, and he's been a wonderful friend and colleague for many years. So Doug, at this time, we'd love to give you your award and hear your comments, please. Please, John. Yes, you hold it. You hold it. I'm going to take a picture here. Here, if you look over there. I should say a word about this, the prize. In addition to the $150,000 prize Hiro and Betty have generously supported, this is the actual piece that Doug is receiving, which has a Waddington landscape at the top, which is meant to signify the concept of cellular reprogramming, building on the notion that Shinya first described, that a cell down in one of these valleys of a cell fate could be pushed back up the hill and be reprogrammed to be pluripotent once again to go down another path. So, Doug.
Thank you so much.
Thank you, Tony. Thank you, Mr. Reyes. Thank you. I can hold this for you.
Yeah,
since it's
that's
so
great.
heavy.
Thank you. Well, thank you, Deepak, for that very generous introduction. And I'd like to begin by saying just how surprised I was when Deepak called me. I didn't know if he was calling me to brag about his recent tennis exploits, but I was delighted and really honored to receive this award. As many of you know, this award is received on behalf of the many students, undergraduates, graduate students, and postdocs who've worked with me over this time, and I'm glad here to represent their work. Before I talk about the work, I did want to say something about Mr. Ogawa. I had the pleasure of meeting him and his wife, Betty, on the extremely happy occasion of John Gurdon and Shinya Yamanaka receiving a Nobel Prize. And I was struck at that time by both their alignment and the intensity of their enthusiasm for science. Having watched the debates, if we call that, on Monday night, and the lack of any sort of discussion about how science does good in the world, it's really wonderful to have people like Mr. Ogawa and his wife who see the power of philanthropy and making lives better for others. And I'm especially honored to receive this award from you and your wife. What I'm going to do today is introduce the disease in which we work, diabetes, summarize some work over the last decade or so that allowed us to get to the point of making a special kind of cell from a stem cell. And then spend the second half of my talk really about things we don't understand and describe our approach, looking forward to what I hope will be a successful conclusion in years ahead, meaning that it will relieve diabetics of pricking their fingers to get a blood sugar test and injecting themselves with insulin. I'm going to begin then by talking in a way about this image here on the bottom left, which is an image of a pancreatic islet. There are about 100,000 to 200,000 of those islets embedded in a person's pancreas. And I'm showing this picture because, of course, the Gladstone Institutes are one of the world's best places for studying heart and other diseases. It's not yet embraced fully my favorite organ, the pancreas, so I just show you this to have you think about it. The pancreas, you see nestled there in the middle of the body, is about the size of a banana. And in that banana then are these 100,000 to 200,000 islets. The islets contain a few thousand cells each, and each of those cells makes a different kind of hormone, the most famous of which, of course, is insulin. So why does this have our attention? Well, there's the pancreas at the top, And you see the two kinds of diabetes, and I'll make the argument here, or at least point out that in both type 1 and type 2 diabetes, the beta cell is a problem. Looking on the right there, you see that insulin resistance in the fat and the muscle and in the liver leads to beta cell dysfunction. So about 10 to 15% of the people who have type 2 or adult onset diabetes have to inject insulin because in effect their beta cells have been worn out by the request for more and more insulin to deal with the insulin resistance. On the left, the disease that has really attracted my attention is beta cell destruction by autoimmunity. And I'd pause here to go back to something Deepak said. Here's a picture of the beta cells disappearing, because I was struck by what Mr. Ogawa said about how one works on these problems as part of a team. And my wife, who couldn't be here today, she may be watching online, really has supported me in this from the very beginning when our son Sam got diabetes. And so I really resonated with what Mr. Ogawa said, that no one takes on projects like this by yourself, and so it's a real team effort, and thanks for bringing up that point. So a quick summary of the history of diabetes then would be this is a disease known for many centuries, but an important event occurred in the beginning of the last century, in the early 1920s, largely based in Canada. Banting and Best and Collip and others, was the discovery of insulin. And so I'll just quickly summarize that for insulin-dependent diabetics, injecting insulin has really been the solution to treating the disease since the 1920s. What I show you in this picture taken from the web, it's a little bit pixelated, but that's a pile, about a 15-foot high pile of pig parts, 2 tons of pig parts. And insulin was extracted from pigs and then sheep and other animals over the years. Those 2 tons of pig parts could give rise to 8 ounces of pure pork insulin, which was at that time enough to inject into about 10,000 people for 6 months. The next major advance in this sort of Whiggish history of the disease was largely based here in the Bay Area. It began with Bill Rudder and others studying this problem. And an important event in 1978 was the production and the cloning of insulin by one of the world's most successful biotech companies, Genentech. I think in part because of that, there's a nice Genentech hall here on this campus. And you see, following that, there were things which were advances, but I would say no quantum leaps forward. It was possible to make human insulin, then fast-acting insulins. Novo Nordisk got the idea of using, instead of a syringe, a pen. Sanofi developed long-acting insulins, but these are largely the same approach to injecting insulin, but by tweaking the protein so that it acted more quickly or had a longer half-life. And I thought I would just show this next slide to kind of set the context for what my colleagues and I are trying to do. So one often hears about companies trying to work on blockbuster drugs that will sell $1 billion a year. Insulin doesn't even qualify in that. It's more like a commodity. It will this year, about $22 billion of insulin will be injected into people the world over. By the year 2019, as it shows here, $32 billion of insulin will be injected. I don't mean in any sense to say that that's not a good idea. That's keeping these people alive and in better health. But what I want to talk about is a different approach. Rather than making more and better insulins and providing them by different devices, to go back to nature's solution, which is the beta cell that makes the insulin. Now, before we get to nature's solution, I do want to point out the advances in providing insulin through pumps. I'm going to just show you some slides I borrowed from Ed Damiano, a colleague at Boston University. On the left, you see blood glucose levels, and for those of you in the back, all you really need to see is that the green bar is the area where the blood sugar level should be maintained, and that's a normal non-diabetic measuring their blood sugar levels in a clinic over a period of 24 hours. And you can see blood sugars, despite eating and exercise, are kept in that range. A compliant type 1 diabetic using an insulin pump and checking their blood sugars would have a scheme more like this, where you see 3 periods of very high blood sugars, 1 period of low blood sugar, and relatively little in the green bar. An advance by Damiano and Steve Russell and others is to make a dual hormone pump connected to a glucometer that measures blood sugar. And there you see on the bottom the improvement over the middle bar, where there's more time spent in the green range. But the point I want to make is that you're still not having good blood glucose control and that you're often out of the green range. And a solution to this problem is to transplant the cells which are absent in diabetics. Here's a picture I borrowed from a colleague, Gordon Weir, who's been a mentor to me. It's a picture of cadaveric islets stained with dithizone, so they're stained red. And these are ready for transplantation. And the point here is that transplanting cadaveric islets has been shown for years to control blood sugars very well. There are two difficulties with this. One is the source of the material, the cadavers, getting enough cadaveric islets in good enough shape to transplant. And secondly, it requires lifelong immunosuppression because obviously the cells come from a person other than the patient. So with that background, I'm going to propose what is really, in some sense, a very simple, almost a pedestrian idea, which is why not make the cells which are missing in the patient? And this has been made possible over years of study of developmental biology, identifying embryonic stem cells and blood stem cells and others. And the important, very important discoveries years ago, which we heard mentioned this morning by John Gurdon and Shinya Yamanaka about the power of stem cells, particularly iPS, or induced pluripotent stem cells. So it's a double pleasure to speak to you today here in front of Shinya, because this is, in a sense, one small version of what I think he imagined would be possible with these cells that he helped devise and develop. So this is, in my lab, a kind of famous diagram. Students are tired of seeing it. They tease me, it's called my Skittle diagram, of what we really have tried to do, which is to say, how do we turn an induced pluripotent or an embryonic stem cell into a beta cell? And it's no surprise to you to realize that that doesn't happen in one step. It's not possible, as far as we can tell so far, to turn this red cell into a glucose-sensing, insulin-secreting cell in one step, but instead it gets educated, the education occurring by what developmental biologists have been studying, which are the genes and the factors that determine cell fate. And so our goal then was to turn these stem cells into this glucose-sensing, insulin-secreting cell. I want to emphasize that the idea is really practically obvious. I mean, if I lecture a lot of undergraduates, and when you talk to them about this, they see immediately that if stem cells exist and if diabetics are missing a beta cell, you don't even really have to ask the question, well, why don't you just turn that stem cell into a beta cell? I'd just say it's easier said than done, but it is a straightforward idea, and I'm going to talk to you about what we've done in a few minutes, and then, as I say, concentrate on next steps. So for many years, we and others, and there are really too many to mention, but I might just mention a few locally here: Julie Snedden, Matthias Hebrook, Gordon Keller in Canada, a couple of biotech companies. Many people have worked on this problem, and it was not so difficult to get to this pancreatic progenitor stage, the green cells, which can give rise to the ducts and the exocrine tissue, which make up about 95% of the pancreas, but also the endocrine tissue. And it was the last steps that proved to be difficult, but we knew we were sort of on the right track, because when we transplanted those human cells into a diabetic mouse, after 3 to 4 months, some of them would mature or differentiate into a functional GSIS, that is glucose-sensing, insulin-producing cell. I think, though I don't know this for sure, that the difficulty in moving from here to here is what inclined the biotech companies who've been working on this to say, well, let's use what we have in hand, which is a pancreatic progenitor or an endocrine progenitor, and see if we can treat humans with that. And I'm pleased to say ViaCyte and Beta-Logics, which have now fused, ViaCyte here in San Diego, largely supported by CIRM, or in large part supported by CIRM, has proceeded to a clinical trial, and we're looking forward to hearing their results of using these pancreatic progenitors in a device. And I'll come back to that later. But this point now is to say, well, that's not the ideal cell, but it at least would be a proof of concept or proof of principle by providing these cells to patients and seeing if they could produce some insulin. We nevertheless set out this goal of, could we really make mature cells in vitro, for 2 reasons. One, it makes more sense to have the cell that you want for transplantation, but also that provides a cell which could be used for drug screening. And I won't talk much about it, but drug screening for type 2 diabetics allows one to try to find out why beta cells become dysfunctional in type 2 diabetes. And so for the first time, one should be able to then screen for drugs on human, not rodent cells, in a large scale. The way we went about this used to take up most of my science talks. Here I'm just going to summarize it in a few slides. One of the reasons we knew we were on the right track was the work of Julie Snedden and others, which showed that by co-culturing these progenitors with endothelial cells, the cells that normally talk to the beta cell, they could be matured in vitro. And so that showed us that the cells we had were had that potential. They were on the right track. We did an enormous number of transcriptional analyses, and here I show a more old-fashioned way of doing it. Now one would do it all by RNA-seq, and combinatorial screening to try to figure out what factors do we need to provide to these cells. And for the developmental biologists in the room, you will not be surprised to know that the important signaling molecules were identified not by discovering any new pathways. All of the pathways listed here, many of you will even work on some of these, all work through a ligand which binds to a receptor, affecting gene expression and controlling the fates of the cells. And let's just point out that in hardly any case is this ever the consequence of a single ligand and a single receptor, but multiple factors being read at the right concentration and the right time. So there, in one sentence, I more or less summarized 10 years of work trying to figure out what is the right concentration and the right time and the right factors. And I show this slide, published in 2014 from our team, which gives you some idea, and I'm not going to go through what the factors are, about how arduous this process was or is. But I want to really concentrate on the fact that we were able then to make these stem cell derived, that is SC beta cells shown here on the right. I'm going to show you a bit more about how we did it, but I'd also like to say at this juncture that in no sense do we believe this process is unique. I'm sure there are a number of ways of doing this. In fact, I'm reminded of Shinya Yamanaka's discovery of the famous Yamanaka factors. People, him and others, have shown there are other ways of doing it. And I'm looking forward to people improving on this by making it shorter and faster and, most importantly, cheaper, because it's quite expensive the way we do it now. Let me show you what is involved. Because of my connection with Gordon Weir when we first started on this, Gordon said something to me which was more or less the following, that it's all well and good for you developmental biologists to show immunofluorescence images of a few cells here and there, but you have to get real. You need to make 1 billion cells per person. And so that got my attention. 1 billion cells is a big number. And so we set out to solve the problem focusing with human pluripotent cells, and to do it at a scale that would allow us eventually to treat people. So rather than doing it in 96 well plates, we did it in these spinner flasks, which I'll show you in a moment, taking advantage of the enormous growth rates of stem cells, which you see there on the right. And these are just some pictures showing that we could get clusters of cells. At 3 days, that sphere of cells is about the same size as an islet, which turns out to be exactly what we want. And then you see some staining for various transcription factors going from the red cell to the purple cell along the process of their differentiation. And what you see here at the bottom is that we can make about 400 million cells in 300 milliliters now in that sort of coffee pot size spinner. But I like to show this slide for a couple of reasons. This slide just shows you in real scale, you'll see a person's hand in a moment, what it looks like. And so in principle then this incubator, which you'll see back in a minute, has 6 flasks in it, which are making enough beta cells for 6 people. Now, of course, this is making them under non-GMP conditions in a way that couldn't be used to treat people, but the numbers are correct. And there you see the little white flecks, perhaps, in the red media are these islet-like clusters. And I like to show this because this movie was made by an undergraduate, Mikey Siegel, who was part of the team. And as Deepak mentioned, I'm not a real doctor, a physician. I teach at an undergraduate college, and my lab has about as many, well, it has more undergraduates than graduate students in it. And so I really like working with a team with undergraduates, and Mikey was a very special one. So what did we do? I'm going to summarize some published work so I can get to new things by saying we are able to make cells which respond to glucose and secrete insulin in a way that's largely indistinguishable from cadaveric islets. So here are adult primary beta cells, islets isolated from a cadaver, challenged with glucose. You can think of this sort of like 3 meals. There's 2 millimolar or 20 millimolar glucose, and at each challenge there's more insulin secreted, and the cells are then depolarized with potassium chloride and secrete insulin. These are very much like the cells that ViaCyte has gone to clinical trials with, and while here they show no response, they have clearly shown that those cells will mature in vitro when transplanted to an animal 3 or 4 months later, and that's what the intention of the clinical trial is. And these are the SC beta cells that we derived with this new protocol. There are lots of ways of showing you that they're very much like adult human beta cells. I'm going to show you my favorite way is just looking by transmission EM. So insulin has been studied for very many years, as has the beta cell, and among its hallmark properties of a human beta cell are these crystallized insulin granules found with a white halo within a vesicle. And they also contain developing or early vesicles here. These stem cell-derived beta cells make identical kinds of granules, whereas the polyhormonal and immature cells make a vesicle that expresses glucagon or has glucagon and insulin and other hormones in it. And so it's not the same as these insulin-only granules. What the cells look like here, this is a great slide to show both the positive and the negative. So on the positive, I'll start. This shows islet-like clusters made in vitro from stem cells, which express insulin with a gene expression pattern that's virtually identical, though not identical, to a human beta cell. There are also a few other alpha glucagon cells here, so that's the positive. And frankly, when we wrote this up and think about it, I tend to only sometimes concentrate too much on the positive. Only 16% of the cells here are what we want. So you could say, well, that's all well and good, but what about the 85% of the cells which you don't want, all those blue cells there? And I'm going to come back to those because that's what we've turned our attention to now. But for the moment, just focusing on the positive, we could make islet-sized clusters, and we could cure virtually any model of a rodent that had diabetes by transplanting these. I'm not going to show you all those because those of you who work in this area will know rodents are relatively easy to cure. You can transplant an insulin pellet and kind of cure them of their diabetes. But here's sort of what the data would look like. If we transplanted the polyhormonal cells like those that have gone into this clinical trial, you just have to take my word for it here that the green here is autofluorescence, so ignore that. This is the transplant where most of the cells are not making insulin, but there are a few bright green insulin-producing cells 2 weeks after transplantation. Whereas if we transplanted our islet-like clusters, you see abundant insulin-positive cells. And interestingly, some alpha or glucagon cells have migrated to the perimeter of these clusters. But there's still plenty of blue cells here that are neither insulin nor glucagon producing. As I said, it's pretty straightforward to cure a mouse of its diabetes. The advance here was using human cells and doing it in less than 2 weeks. And these cells respond very well, as do human cadaveric islets, to a glucose challenge inside of the mouse. So if we go back to a blow-up of this, this is our challenge then. How do we get better control? I sometimes call it like mastery and dominion over these cells to tell them to do exactly what we want. And how do we then move to deal with the problem of the autoimmune attack? And so I'm going to spend the rest of my time talking about those two main problems. So to summarize what I've told you so far, by comparing these SC or stem cell-derived beta cells to human islets, on gene expression, insulin granules, I didn't show you intracellular calcium flux, making fenestra with endothelial cells, multiple GSIS or glucose challenges in vitro, and rescuing, rapid rescue in a mice. These cells are looking very good. What are the problems? I would say efficiency in a number of senses, and I want to concentrate a bit on controlling the cell type and then finish up on their immunogenicity. So here's one of the best examples we have of comparing in a dynamic way how our SC beta cell clusters respond to glucose. So again, here are human cadaveric islets. The gray bar is very low glucose. The blue is low at 2 millimolar or 2.5, then high glucose challenge, and then again potassium chloride for depolarization. And this is what a good islet or beta cell should look like. These are the cells that are now in human clinical trials, but again, I want to emphasize they are going to mature after 3 or 4 months in a mouse and hopefully in a person. These are the cells we've made, and this is frankly one of the very best examples we ever have. And I want to point out two things. One is their spike of insulin at high glucose is lower. That's 0.8 here compared to, say, 3 here. And secondly, if they respond at all to low glucose, they don't respond properly. So we haven't really got it down so that they are working perfectly in vitro. They do work perfectly in vivo after transplantation, but we really want to focus in on their in vitro functional characteristics, and Jeff Davis and Ronnie Hellman are very active on that. In terms of controlling insulin secretion in vivo, many of you will know, of course, that the islet contains more than just the beta cell. And so I want to look ahead now, admitting that we haven't crossed the goal line on the beta cell. We're maybe in the red zone or near the goal line or something. We pretty much have a good idea of how to make the beta cell, but I want to focus on these other cell types. In particular, we can now make alpha cells, as others have done, and I won't show you that data. We are ignoring pancreatic polypeptide because no one really seems to know what it does. And ghrelin is important for appetite, and diabetics in general don't have trouble with appetite. But the delta cell, the somatostatin-producing cell, is a very interesting cell because it keeps beta cells happy and healthy in mice. If you remove delta cells, beta cells do not do well. And more importantly, they're involved in shutting down insulin secretion, so you get those nice spikes rather than a bolus of insulin, then a long trail. So I'll quickly summarize the work of Quinn Peterson here, who by modifying the protocol has now been able to make delta-like cells. So here's the protocol. The point here would be looking at somatostatin in green, and then PAC6 is a good marker for the delta cell. And if you look again down here, delta cells do not express the transcription factor NCAC 6.1, but here you see 45% of the cells making somatostatin. But C-peptide or insulin is also made in these clusters. Again, one of my favorite assays, the transmission EM, shows a little hard to see here, but here's a delta cell, and here's a stem cell-derived delta cell. So we can make these cells now, and we're struggling, and maybe some of the endocrinologists here can help us with this, to figure out how we would really prove their delta cells in vitro. There's not much work done on them because there aren't many of them. No one's ever had them in pure form. And so we found that they respond to glucose, but in the way opposite that a beta cell does. So at high glucose, they secrete less somatostatin, and they secrete more at low glucose, and they, well, of course, when depolarized with potassium, secrete somatostatin. We're just learning about delta cells, and I'd be glad for advice on that. If I go back a few slides to this one, onto this issue of if we want to have really perfect physiological control in the transplantation experiments, looking ahead to humans, of course, we want to get better control over how we make the beta cell, the delta, and the alpha cell. And so I'm going to spend a fair amount of time now on how we've approached that. I don't know that it's the only way to do it, but I'll just tell you how we've done it. So the goal here now is to say, can we learn enough about the genes and the factors so that we can tell every cell in our spinner flasks what to do? The first thing we did is by just looking at the fact that there are synchronized switches in gene expression. This is looking, it's an expensive experiment, doing 14 different flasks all separately and looking at wholesale gene expression of all of the cells by RNA-seq. And what you can see here is the cells start off pretty uniformly, but by the end, there are cells expressing insulin rather early. Doesn't really matter what the markers are. The point here of this experiment is to show that the cells are not moving in lockstep. They're not like a marching band where all of the cells respond at the same time in the same way. This nevertheless allowed us to find new genes, genes not otherwise studied, which either go from on to off or off to on at these various stages. But even that doesn't really help if you want to focus in on single cells because you have 300 to 400 million cells in the flask. So when you do this analysis, you don't know if you're looking at two genes, if they're expressed in the same cell or not. So fortunately for us, Adrian Vera, a graduate student in the lab, was involved in these recent papers known to many of you on developing methods for single-cell sequencing, and that's what I'm going to concentrate on now in the next few minutes. So I'll show you this little movie here. This is a very clever method which allows one to mix a bar-coated hydrogel with a single cell at a high rate and then do batch sequencing, but yet deconvolute, if you like, the sequences to know which genes which were expressed in which cell. And one way I like to think about that is instead of now doing a flask, as I said, we did 14 flasks, each with hundreds of millions of cells. We've now done 50,000 independent experiments, sequencing 50,000 cells at all the stages, and then look at that data and say, if we now look at 50,000 experiments, can we figure out what's going on? So that's the puzzle. If you have this RNA-seq data from 50,000 cells at all stages of the process, can you work backwards and figure it out? Sometimes when I describe this to undergraduates, I say it's sort of like you came to this country and you didn't know about football, and you were shown still pictures of a football game, like someone passing, someone catching, someone tackling, and you tried to figure out the rules of the game. It'd be pretty hard to do unless you were watching it with an American live and he was explaining to you or she why we have a 10-yard line, something like that. I don't know if that's a good analogy, but that's sort of like the puzzle we have. So I'm going to show you where we are with this, because I think this approach is going to work, and it's actually already got us quite far along. Here again, we go back to the skittle diagram, and one of the first stages at which things go wrong, you could say, is when the green pancreatic progenitor cells, cells which can make any part of the pancreas, start to diverge at a significant level. So here's a FACS plot where we know that the cells we want are expressing NKX6.1, a transcription factor, but a lot of the cells already start to express chromogranin. You see that at the 23% in the bottom right. And that's too early to express chromogranin. We don't want them to be doing that, but they do it anyway. So the challenge is, how do we identify all the cells in there and then figure out why they're listening or reacting to the wrong factors? Here's maybe a more pertinent example at the end of the process. So I've emphasized a few times that the cells we want are making insulin, of course. They're also making this transcription factor NKX6.1. And so in this flask, 19.5% of the cells are what we want. But what are all these other cells? Why didn't they get the message or the memo? Why did they get stuck here? Why did they go there or here? We're trying to figure that out. And some of you will know how this data looks, but since it's a relatively new way of presenting data, I'm going to show you 18-dimensional data on all of these 50,000 cells, reduced to 2 dimensions with just a color code. So I asked Adrian to prepare this slide for me because I wasn't trained in math and have a hard time with t-distributed stochastic neighbor embedding, which is how these graphs look. And this just explains what you're going to be seeing. If you look at cell 1 and cell 2, at these genes, they're very similar in their expression. There's insulin and MAFA, a little bit more on in cell 2 than in cell 1, but these other genes, they're very similar. Similarly, cell 3 and cell 4 express these genes in a very comparable way. But cell 1 and 3 are quite different, as are cell 2 and 4. So in the data I'll show you, it presents data that have similar gene expression of single cells in 2 dimensions. So if we look at that first stage when cells start to go awry, this sort of mushroom-like diagram is single cells where the shading intensity on every single cell shows the level of expression, in this case of the NKX6.1 gene. If we look at other genes, we can immediately see then that these polyhormonal cells, which we don't want, are segregated off, and we can further find that the bridge in between them there, the cells are expressing neurogenin. So we begin to make sense of what's happening to these cells and when. I'm going to go just to the end stage, what we call stage 6, and describe the cell populations which we've identified now by this single cell analysis. And if you look here at the diagram of a stage 6 flask, and Adrian purposely used one that didn't have an exceptionally high success rate because he wanted to collect all the kinds of cells which are, you could say, the troublemakers. Here you see where insulin is expressed and MAFA, a transcription factor marking very mature cells. This analysis brought to our attention many transcription factors and transmembrane proteins. One I'm showing here, ROBO2, not really previously attended to by beta cell biologists. This turns out to be a very good surface marker for purifying these mature beta cells. And so we're using that kind of information to know which receptors are on the cells and also to think about how to use that, say, with a magnetic antibody system to purify the cells. If we look further at this data, we can see the cells which are on their way to becoming these mature cells, expressing these transcription factors. We also then find these polyhormonal cells, these what I'd call troublesome cells. They kind of turned down the wrong alley and got stuck. And then we also find some cells which, as far as I know, do not exist in, I don't want to say in nature, but let's just say in a normal pancreas. They're replicating cells expressing an odd set of genes. Again, they seem to have, it's kind of interesting that you can make a stable cell that doesn't exist normally. If they exist normally in development, they may be killed off. We wouldn't know that. But these two red and kind of green cells are troublesome, let's call them. So what do we do with that information? Well, this allows us to make this following kind of lineage diagram now. And there are a lot of different kinds of cells on here, and you could say, well, this is just getting worse, not better. It's getting more complicated. But actually, it's still the purple cell that we want to make, going from the green cell. We now have a very detailed idea of which genes those cells express while they're on their way to the purple cell. And we also have a very good idea of the genes that are turned on when they take what we would call the wrong path. I could show you a similar kind of diagram for the delta cell and the alpha cell, but I think you get the idea that we're trying to figure out how cells make decisions about their fate and when do they make that decision. So I'm going to finish this part by telling you our program of how we're going to try to get control over this. The first way is to study in detail all the receptors expressed on the cells at these stages, and then use ligands for those receptors to try to skew or force them down the right path, down the right road. But Adrian and others are taking a different approach at the same time, which has been made possible by the enormous advances that the whole community of biomedical scientists have created, which is really more information about sequencing, the development of CRISPR technologies, the development of institutes and core facilities that allow investigators to do experiments which were just years ago sort of inconceivable. So in this experiment, we're making use of the Broad Institute's RNA guide library, which has over 100,000 guide RNAs, 5 for each human gene, into lentiviruses to infect a pool of cells. The other important advance here is that we can make, as I mentioned and maybe emphasized, 500 million cells at a time. So this is not an experiment you can do in a tissue culture plate. You need to have hundreds of millions of cells in order to do this experiment. We infect the cells at the embryonic stem cell stage with this virus pool, and then proceed with the differentiation and sample out cells by fax of all the different types we get and sequence them. Because we're knocking down one gene at a time, we can then ask which gene do you have to knock out to either prevent them from going down this alley or encouraging them to go more vigorously towards the right cell type. So this is what that experiment will look like. So now we have a pretty good idea of the kinds of genes we want to do to control it. This is not a result. This is the goal, okay? So the goal is now to infect pre-beta cells or embryonic stem cells with guide RNAs that knock out transcription factors that are required for the cells to go down the dark alley. Essentially, we're going to put blinders on them and say the only thing you can become is a beta cell. I don't know if this will work, but this is our favorite idea at the moment, and this is where we're putting a considerable amount of our resources. If that works, we'll do the same thing for the delta and the alpha cell. And then to kind of dream ahead, the goal is that we will be able to custom order an islet. You'll be able to say, how many beta, alpha, and delta cells do you want? And we'll give you those in that proportion. And they'll be human cells. I'm going to finish by talking again about the future more than the present. And that is, if we have solved the problem of making an infinite supply of glucose-sensing, insulin-secreting cells, for the type of diabetes that interests me, we have to solve the autoimmune problem. There are two ways to do that. One is to sort of give up on the biology, say immunology and autoimmunity is so complicated we'll never understand it, so we give up and we'll just make a moat. We'll just physically encapsulate the cells to protect them. I'm going to talk a bit about that, but we're also going to pursue the sort of biological approaches to say, let's try to figure out on the biological side what we could do to make the cells opaque, or if you like, immune to the autoimmunity. And there, working with Chad Cohen, we've done many, I'd almost say obvious things based on the work people in this community and others of using CRISPR to knock out the sort of billboards, the histocompatibility locus, to knock in PD-L1, CTLA-4 Ig, others into these cells to say, can we make a cell which will blunt, if not somehow make the cells tolerant? I'm not going to talk about that. I'm going to instead talk about encapsulation. This, as I say, is a sort of more bioengineering approach. As Deepak indicated, I'm not a bioengineer. I barely got through organic chemistry as an undergraduate. But luckily for me, I work in Boston where there are lots of bioengineers. And among those we work with, we've enjoyed a really great collaboration with Dan Anderson at MIT. And so he would be annoyed that I'm going to summarize about 15 years of his work with lots of postdocs in one slide, which he kindly gave me. The idea here, let me go back to this slide to show you what the idea is. The idea is to encapsulate our human islet clusters in alginate so that glucose can go in and insulin and hormones can come out, but immune cells can't get in nor kill the cells. And so Dan Anderson and his colleagues began with alginate up there on the top and did an enormous amount of novel material design, chemical modifications of the alginate, then tested them by a combination of adding them at different times and places, as it were, analyzed the material, developed islet encapsulation, did what he jokingly calls a high-throughput mouse, where he's looking at the amount of immune attack on the naked capsules by putting, as you see here, 8 different capsules in a mouse at once. And I'm going to show you the results where we now use his best design of what's called an RZ15 modification of alginate, putting our human islet clusters into a mouse that has a fully active immune system and is diabetic. And so the good result here is that if we put in blank capsules, it does not cure this animal that's been treated with streptozotocin, so it has a high blood sugar. A healthy mouse, this is in green, its blood sugar level. And here's just 250 stem cell clusters with this modified alginate now out more than 190 days, living, if you will, perfectly well with human islets in the presence of a fully active immune system. Now, before one jumps ahead and say, well, that's great, just go ahead to human clinical trials with this, this has the problem that this would involve putting hundreds of thousands of these capsules into the peritoneum. The FDA and the patients are not going to like that. We have to figure out ways of how to present them, and that hasn't been done yet. And we're also trying, through a company I'll mention in a moment, to use a different kind of encapsulation device, a macro device to protect the cells. But I want to end with 2 slides here. One is to just show you, in a sense, a kind of vision slide, that I believe that the future of diabetes, going from the discovery of insulin to the injection of insulin, is now at an inflection point where we can think seriously about patients not injecting themselves with insulin and not having to prick their finger to check their blood 10 times a day. On the left, you see a diabetic who has high blood sugars, and on the right, one with normal blood sugars, which I think can be done with this straightforward, sort of simple-minded, I would call it nature's solution to the problem. I do not want to underestimate or in any way de-emphasize the amount of work done by so many other people that makes this kind of approach possible. Many here, as I've emphasized, in the Bay Area. I like to think Boston is at least half as good a place to do science. And it takes a community of people to really solve a big problem like this. In my own community, it's the lab where I work, and I'm so grateful to all the people who've worked in it in the past. Here I'm showing you mostly present people. This includes, as I've said, undergraduates, graduate students, postdocs. And we also enjoy wonderful collaborations. I've mentioned Gordon Weir, Dale Greiner and Lenny Schultz help us with these humanized mice for testing our cells. I mentioned Dan Anderson. We're working now with Mark Poznansky on a different kind of encapsulation device. People from the biotech company I'm involved in founding, Sema Therapeutics, George, Poe, and Felicia, we work closely with them. And for disclosure and thanks, I want to point out how this work is funded. In our own case, because of the reasons Deepak mentioned early, much of this work was done almost entirely with philanthropy because it began at a time when our nation doubted that federal funds should be used for this kind of work. And that was provided by the Harvard Stem Cell Institute, the Howard Hughes Medical Institute, the JDRF, the Helmsley Charitable Trust, the JPB Foundation, and the NIH. And we also now have a collaboration with AstraZeneca on the in vitro analysis of these human beta cells. So I end with thanks in two ways. One, to the people in my lab, and secondly, again, thank you to Shinya and Mr. Ogawa for this honor, which I'm so grateful to receive. Thank you all.
Thank you, Doug. We do have some time for questions from the audience and our online viewers. Yes, Melanie?
Great talk. I wanted to know what your estimate is how long the S beta cells will function in a patient in the capsules. Do you think that this will be something that is lifelong or will have to be renewed? And secondly, where do you think these capsules should go? Is it going to matter to keep certain factors at a high concentrations to keep them in their face, or can they go anywhere in the body?
Right. These are great questions, which I might just repeat. It's about the longevity of the cells and where they would be put. All I can say about the longevity, you asked me kindly, what do I think, rather than what do I know? So I'm just going to tell you what I think, because we don't know yet. They've lasted, as I've said, more than nearly 200 days in a mouse. I don't know what we can conclude from that, but they've lasted a very long time. I think because the cells are in some sense like fetal cells, they're young embryonic cells, they're likely to last a very long time in humans. So what does long mean? My children ask me that. What is long? I don't mean a week or a month. I would be very surprised if they did not last years. But that's just based on intuition, not on any fact. As far as where they would go, I actually believe the first clinical trials won't be with those small capsules, but will instead be with a device, the kind of device that Sema Therapeutics is developing, and the one that and the Theracyte device, as it's called, that ViaCyte uses. That is a good idea in the sense that it's easily retrieved. If you put thousands of capsules in the peritoneum, you could try to vacuum them out, but you're going to miss some. This is a retrievable device. So I think that will be the first step. Your question reminds me that I neglected to say that we're also trying, and this probably requires too much explanation, to go into a trial into patients that do not have autoimmunity. And so there we're going to just try to solve one problem. These are insulin-dependent, non-autoimmune diabetics, people that have either had a pancreatectomy or for other reasons require insulin. And so that will allow us a kind of proof that the cells are as good as I've claimed they are. So the cells, we're very confident with the cells. How would you put it? If you were a mouse, we have really good news. We can give you human cells. But we haven't done large animals and we haven't done humans. So we want to go kind of in a stepwise way. Did that answer the question?
Doug, along those lines of the selective populations, can you say something about the potential use of autologous cells that you talked about earlier?
Yeah, thank you, Deepak. We're doing 2 autologous programs with iPS cells. One here in California, supported by CERM, and that will involve taking blood from 10 to 20 patients, turning their cells a la Yamanaka into an iPS cell, turning those cells into beta cells, and then putting them back into those patients because these are, quote, the patient's own cells. And so they not only don't have autoimmunity, but there shouldn't be an autoimmune rejection. We're doing the same kind of approach with a different patient population in Boston. And it's always worth noting that this doesn't ever happen fast enough. If everything worked perfectly, which in my life it never has, that could be in 3 years that they would go into patients.
Bruce?
So I was intrigued by all the different cell types because they seem to have proliferated since I last studied that you're studying. And to what extent do you think that diabetes is an insulin deficiency versus a somatostatin deficiency versus a glucagon deficiency, especially given the results of the dual, my understanding, I guess, is that the dual pump is actually secreting glucagon as well as insulin.
Right. So as you know, the glucagon is there to prevent low blood sugar by causing the liver through gluconeogenesis to make more glucose. So the reason you put glucagon there is you don't want to overshoot with the insulin. You asked a deeper question in a sense, which is the disease really a beta cell deficiency? So what I can tell you is that in type 1 diabetics, the cell that is missing is a beta cell. In fact, there's an alpha cell proliferation, and there's good evidence for delta cells. So there's no reason to believe that the other cells alone are sufficient to control glucose homeostasis. They're not. But I like your question because I do believe that those cells didn't evolve, nor are they all put in the islet together by accident. And so I think the crosstalk between them is going to be required to get perfect control. But let's remember, medicine's goal shouldn't be perfection. I want to get control that's more convenient and better than injecting yourself with insulin. It reminds me also to comment a bit about the earlier question about where we put the cells. One good thing about this disease, say unlike more complicated neurological diseases where you probably have to replace brain cells in the brain, you can inject insulin pretty much anywhere, in your tummy and your, whatever the proper word for butt is, you can put it anywhere and it'll work. And so we're not so worried about where they need a good blood supply, but we don't have to put them back into the pancreas.
We have one question here and then here. What's that?
Can mammals endogenously regenerate islets, and is there a developmental or postnatal window where that occurs, and can we learn anything from that to educate creation of cell therapies?
Yeah, it's a good question about whether there's any regenerative capacity in the beta cell. We showed many years ago that beta cells derive from pre-existing beta cells. So once you remove all of them, in my view, the capacity to make more is either zero or so near zero as to be physiologically zero. There is evidence in mice that under certain transgenic conditions, if you kill beta cells in a certain way, some alpha cells can convert to beta cells. I think Deepak and others, Marius Vernig was mentioned, I know Xin Ding here has worked on this, have been working on this idea of, let's call it transdifferentiation or conversion. I think that has real clinical potential, but your question was, does that happen in vivo? And I think rarely, if ever, and if it does, it doesn't happen at a level that's physiologically significant. Is that good? Yeah.
Casey?
You actually just sort of answered my question. So, given your work on in vivo direct conversion, because you have done a little bit of that, even though you haven't talked about it, I was wondering why you moved forward with IPS-based therapies as opposed to choosing a direct conversion therapeutic.
Yeah, well, thank you, Casey. I mean, I think logically one could follow on our work and others, that of Zhou Zhao and Xin Ding and others, and say, let's convert exocrine cells or other cells into beta cells in vivo. I don't think that's a bad idea. I guess my kind of weak answer is I focused on this because we can make so many cells, and we're pretty far along in making beta cells this way. I put a huge advantage to being unlimited in the starting material. The more we do, I mean, if you think about that complicated gene knockout with all the guide RNAs, that just requires lots of cells. So it's not a good scientific answer. It's more like my intuition is this is where I'll place my bet. But if you want to try the other thing, go for it. Yeah.
Casey's question reminds me that there are many here on campus who benefited from Doug's mentoring, including several of our faculty, and Casey, for whom Doug served on her thesis committee as a Harvard PhD student. Lisa?
Hi, kind of going back to Deepak's question. That was an amazing talk, by the way. Thank you so much. If the cells are already encapsulated, is there any reason to not go with an allogenic as opposed to an autologous approach?
No, I should have emphasized that. The approaches I talked about with the encapsulation are all allogenic. In fact, Deepak and I were chatting this morning about the challenge for biomedicine is autologous makes such good sense. There's no reason not to do that except for cost. And so the cost we're looking at now for our autologous program, believe it or not, are about $1 million per patient. But I think if we demonstrate efficacy that way, the numbers can come down. And then I often turn it around to say, if you had diabetes and you wanted to free yourself of injections and blood glucose checks, how much would you be willing, or should your insurance company be willing to pay to relieve you of that and eliminate the complications due to the disease, not to mention change your lifestyle so you can eat what you want, play as much tennis as you want, and not have to worry about monitoring your blood sugar all the time. But yeah, thank you for that question. So the program we're on now and with encapsulation is focused on allo cells, one universal bank of donor cell that will be put into the device.
Question here, and then Jensen.
Wonderful talk. I was just curious about the fact that it's been published recently that there are multiple subtypes within beta cells, that they're perhaps not a homogeneous population as we once thought. Do you see this in your single cell analysis, and do you think this might have some importance in tweaking the glucose responsiveness of your stem cells?
Yes, that's a great question. It reminds me of two words I used loosely today. One was a mature beta cell. You should think when I said mature beta cell, I mean that in the same way that, like, who in this audience is mature, right? All right, so like we're all at different levels of maturity, and I'm sure beta cells are like that as well. I think one should be careful in interpreting those papers to mean whether there's any stable different state among the cells, or whether you're looking at a physiological difference because you've got a snapshot of the cells. So a beta cell that is just divided will probably express different genes from one that hasn't divided for 10 years. A beta cell that has just responded to a glucose challenge and secreted a ton of insulin will also express different genes than one that hasn't responded. And when you have a meal, you probably know all of your beta cells don't respond, only a small percentage do. So that's a long way of saying that those are interesting papers on the possibility of heterogeneity. Heterogeneity wouldn't surprise me. We see heterogeneity in the beta cells we make. But I think we have to focus on functional heterogeneity. And so it's the functional test that we really care about.
Jensen, you had a question?
My question is about pancreatic malignancies. Do you think your work on making beta cells, one cell type in the pancreas, teach us anything about the morbidity and mortality associated with pancreatic cancers?
Yeah, it's an important question. I often say, not in a joking way, that I don't like to think about cancer. We don't really study pancreatic cancer. Some of my former students, notably Ben Stenger, is working a lot on that. What I can say is that the evidence suggests that the pancreatic progenitor for the cancerous cell likely arrives from that sort of middle of the process I showed, the green cell, not from any of these later stages. We don't see any pancreatic cancers when we transplant our cells. And so I don't know, and I don't think it is known, what is if there is a source of a pancreatic cancer, like if there's a stem cell, if there are multiple ways of getting there. But we frankly don't study that much. But you've implied with your question, why doesn't one use single cell sequencing to try to figure out where those cells come from? And I think that's a good idea. We're not going to do that. Yeah.
Dana?
We have a question for you from your online audience. First, Jennifer Hugh, who's a former undergrad in your house at Harvard, says hi and congratulations. And then also we have a question, other than autoimmunity, is there any difference in treatment of type 1 and type 2 diabetes with beta cells?
Yes, it's a good question. Thank you, Jennifer, for still paying attention. There's a big difference in that type 2 diabetes is a metabolic disease that unquestionably involves a lot of lifestyle issues, diet and exercise. That's not true for type 1 diabetes. It doesn't matter diet or exercise. It's, the autoimmunity is a different cause of the beta cell loss and dysfunction. So the reason I combined the two today is about 10 to 15% of type 2 diabetics inject insulin. And so what I talked about today is relevant to them. But other endocrinologists here will be better positioned to talk about how type 2 diabetes should otherwise be treated, which at a first pass is with diet and exercise.
Well, in keeping with the theme of it takes a team, let me end with thanking Hiro Ogawa again and Shinya Yamanaka and our team at Gladstone that has helped make this event possible, including Megan McDevitt, Eleanor Prezant, Dana Smith, and Ann Carollo. And most importantly, thank our awardee, Doug Melton, for a wonderful lecture.
Thank you. Thank you. Thank you. Thanks, Deepak. I see that. I'm going to have to figure out how to...
Oh, my.